Alright, so this is my web diagram that I made. I started in the middle with our different types of models, the sandwich model and the fluid mosaic model, and they're made up with the plasma membrane, and then later on we go into more detail about the phospholipid bilayer, which is composed of the hydrophilic head and the hydrophobic tail. The hydrophobic tail has the saturated and unsaturated fatty acids, and then inside and around the phospholipid bilayer we have membrane channels, membrane proteins, and these can include receptor, anchor, and enzyme proteins, transporter proteins, integral and peripheral membrane proteins. Alright, and then so over here for other membrane proteins we also have, these proteins generally will bind or allow things to move in and out of the cell. These proteins, enzyme proteins, will bind with an enzyme and allow things in and out of the cell. Anchor proteins and receptor proteins are not really used for diffusion. And then these are different types of proteins. So monolayer, they're only in one layer. The lipid linked, they are linked together in the lipid layer of the phospholipid bilayer with covalent bonds. And then protein attached associated proteins, those are going to be linked with the proteins that are going to be inside of the phospholipid bilayer, but they're linked with very weak non-covalent bonds. The plasma membrane can pinch off to give us vesicles, so kind of like if this closed off right here and then popped over here to create another little cloud. And then I went up here and showed how this model of the plasma membrane and phospholipid bilayer shows us selective permeability. That is also has to do with simple diffusion and osmosis. Osmosis is the flowing of water through a selective permeable membrane in order to go to areas from high concentration to areas of low concentration, so the concentration gradient. And that kind of goes with the electrochemical gradient, because if this is our membrane of the cell, or this might be easier to see, if this is the membrane of our cell and we're in a very salty solution and it doesn't match what's the cytosol on the inside of the cell, water is going to go out of the cell to try and neutralize this environment. And so it will go down, up or down its electrochemical gradient. And this can be done through passive transport and active transport. Passive transport is when it will go down its natural electrochemical gradient. Active transport utilizes ATP and energy in order to activate these membrane proteins and these kind of proteins up here in order to allow the movement of molecules in and out of the cell. And this is done through these ion channels. And these ion channels are proteins that are also located within the plasma membrane. There can be the gated ion channel, which, like I was saying up here, has to be opened with energy in order for a molecule to pass through, and the channel mediated passive transport. So passive transport does not require energy.